Welcome students of grade 8 and peace be upon you. Today we're going to talk about a process that occurs during the inflammatory action, which is the second line of defense, called phagocytosis. The objectives of our lesson are: so let's make a quick revision. After crossing the natural barriers, an inflammatory action takes place where leukocytes fight pathogens. During an inflammatory action, which is the second line of defense, the characteristics of the inflammatory action are rapid, local, protective, and non-specific. The inflammatory action is a non-specific immune response since it acts the same no matter what the pathogen is. The signs of the inflammatory action are redness, swelling, warmth, and pain, and we talked about the reasons of each sign. After infection, some cells that are monocytes and granulocytes leak into the site of infection. But what is its role? They will eat and kill pathogens by phagocytosis. But what's phagocytosis? Let's watch a video to explain phagocytosis, its role, and way of action. This is a macrophage destroying pathogens in an infected tissue. But when does it happen? Which cells would undergo phagocytosis? Let's find out. Then, phagocytosis is a process that occurs during inflammatory action. The origin of the word phagocytosis is Greek, hence phago refers to eat and cytosis refers to cell. What are the cells that will undergo phagocytosis? Phagocytosis is done by two types of white blood cells that are monocytes that will be changed into macrophage while passing into tissues, and the granulocytes. Then, since these two cells only can perform phagocytosis, they are named phagocytes. Then, what are phagocytes? Phagocytes are the cells that will perform phagocytosis and are monocytes and granulocytes only. So, let's watch a video showing a granulocyte performing the process phagocytosis. As we can see here, the granulocyte approaches or comes near to the pathogens. Then it sticks to them and let them inside it. Then it cuts them into pieces and kills them. But this is not enough. Let's divide this process, phagocytosis, into steps and explain each one separately. So, the first step of phagocytosis is approaching of phagocyte to the microbe. Here we can see the granulocyte detecting the microbes and coming close to it. The second step is adherence. Here the phagocyte or the granulocyte adheres the microbe or sticks to it by deadly chemicals. Then adheres means sticks. The third step is surrounding. Here we can see the phagocyte surrounding the microbe progressively. After surrounding, the fourth step is engulfment. So the phagocyte engulfs the microbes and takes them inside it. After engulfment, digestion starts, by which the phagocyte, which is the macrophage or the granulocyte, digests the microbes and destroys it. After the microbes are digested by the phagocyte, elimination starts, by which the phagocyte eliminates or gets rid of the destroyed microbes. So, let's summarize all these steps of phagocytosis in one complete process. First, The phagocyte, granulocyte, or macrophage approaches to the pathogens and adheres or sticks to it. After that, it surrounds the pathogens and engulfs them to its inside, introducing them to be digested by chemicals. Finally, after digestion, the microbes will be eliminated to outside the phagocyte by elimination. As a summary, phagocytosis is a process during inflammatory action. The phagocytes or the effector cells are monocytes and granulocytes. The role of phagocytosis is to eliminate pathogens regardless of their identity or shape, and it's non-specific. Also, this figure shows the steps of phagocytosis. So, let's describe each step and give it a title. The first step is approaching of a phagocyte to a microbe. The second step is adherence. The third step is surrounding. The fourth step is engulfment. The fifth step is digestion. And the last step is elimination. So, as a result, after the first natural barrier, which is the skin, is crossed, the pathogens enter and they start to infect cells. Then the pathogens damage some cells, and the damaged cells secrete chemicals that causes the increase in the blood flow to the wounded area. Also, the chemicals causes the surrounding capillaries to dilate and leak macrophages and platelets into the wound. Then, the macrophages that has squeezed into the site of infection start to engulf bacteria as well as dead cells, and this is phagocytosis. As shown here, we can see macrophages engulfing some bacteria by phagocytosis. And at last, platelets that moved out the capillaries seals the wound and stops bleeding. As an evaluation, please pause the video to solve this application. The following figures show 3 different steps of phagocytosis done by a phagocyte. Please identify the phagocyte and give a title to each step and justify. Before we start, we need to identify the phagocyte. A phagocyte may be a granulocyte or a monocyte. But how can we distinguish between them? Excellent, according to the shape of a nucleus. Here is a cell having a horseshoe-shaped nucleus, hence it's a monocyte. And here's a cell having a multi-lobed nucleus, hence it's a granulocyte. So let's now identify the phagocyte in this figure. Excellent, this phagocyte has a horseshoe-shaped nucleus, hence it's a monocyte. But what's the title of each step? The first step shows the sticking of bacteria to the phagocyte, hence its adherence. The second figure shows the phagocyte surrounding the bacteria progressively, hence its surrounding. And the last step shows the phagocyte digesting the microbe and destroying it, hence its digestion. But what would happen if the phagocytes failed at defending the body and killing pathogens? Let's find out the next time. This is all. Hope you enjoyed the lesson. Study well and keep safe.